Testing, testing. Is anybody out there? Hello? Started a few minutes early. Hi guys. Fantastic. I forgot I had that scroll. Brandon, did I tell you I have a scroll? He's got headphones on. He can't hear me. That's my problem. I have a funny it's scrolling thing. My husband is currently cleaning the kitchen, so if you hear a bunch of stuff about 8 feet away from me, there's a reason why. But the good news is that the kitchen looks beautiful. He can't hear me, but it looks great. I miss you guys. I'm sorry that this looks like the rest of our semester. Stephen, you waited a whole semester for biochemistry, and this is what you get. Okay. We'll have one more thing. In case you don't remember, you can, advising registration started today. So if you have not emailed your advisor yet, you should probably do that. Oh, that's not what I wanted to do. I'm glad you guys got my email. Fantastic. It'll be great to see you in the fall, hopefully. Okay. Let's see who else with us. YouTube is not receiving enough video to maintain smooth streaming. As such, viewers will experience buffering. I don't know how to fix that, but hopefully we'll make it work. Today's easy anyway. Drew, aren't you my advisee? Have we done advising yet? We should probably do that. All right. So today, in this edition of biochemistry, we're going to be learning some new words. They're words that you might already know, but it's one of those things where I kind of have to act like you don't know them because some people might not. And so we need to go over them, and as a community of class, like our class, I need to make sure that we know these words and that I can use them and that you know, everything kind of makes sense. So the easiest way for me to do that today is to write down words. Oh, I hate a white screen. Let me do a thing. Yes, you need to get on that, sir. Okay. My coworkers are officially four-legged animals. So if you hear pitter-pattering behind me or anything, I apologize. That's just what we're doing now. All right. So to do this type of streaming, I kind of need to have, let's see, I have 1, 2, 3, 4, 5, 6. I have 6 different windows open. And so it takes a lot of RAM, and it takes a lot for me to click between screens sometimes. So if I don't happen to have my chat box up at any given time, I might not see that you're trying to get in touch with me. So if that's the case, I'm sorry. I try and keep it up though, so that way I don't know if you're asking questions, but it doesn't always work as well as I want it to. But this is what we've got, so. Okay. I'm going to start off today's lecture with a couple of questions. Why do we eat? I think it's a valid question. Not everything eats. Plants don't eat. Hey, Brandon. What's he doing in there? Let me mute this real quick so I can yell at him. Hey, Brandon. Can you keep it down a little bit?
I'm sorry.
It's okay. He's wearing headphones. He can't hear me at all. He has no idea how loud he is right now. Okay. So the question, again, why do we eat? So plants don't necessarily eat. There's some carnivorous plants that do eat. Oh my gosh, why are you guys so loud right now? I can see that this is going to be annoying really quick. Anyway, we have to eat because we need energy. That's why plants don't need to eat, right? Because they don't need to get energy. They make their own through photosynthesis. Hold on just a second. No, and all the dogs are being Can you like take her bell off? Thank you. Let's try this again. So sorry. Okay. So why do we need eat? We need energy. And then why do we need energy? I'm giving pause for tea, but I'm hoping you're thinking about these things. But really, we're having to make things that we don't eat, right? So we've kind of talked about this concept of biological molecules. We've talked about it in a couple different capacities. But I think it bears repeating. So like, let's think about the different types of biomolecules that we can kind of think of and hear about. So we have the nucleotides or nucleic acids. So DNA and RNA. We've got things like peptides, polypeptides, amino acids, right? Protein and protein adjacent type building blocks. We've got sugars or saccharides. So monosaccharides, disaccharides, polysaccharides. We've got all of those minerals that we talked about earlier on in the semester. So like calcium ions, iron, obviously we use that in our blood. We have a bunch of these different minerals that are basically floating through our bloodstreams and in many cases attached to our proteins as specific metal cofactors. We also have those coenzymes. So if you remember that word, those are those organic cofactors. Things like ATP, ADP, NADH, a lot of the ones at biotin, niacin. You've heard of a lot of these because they're derived from dietary vitamins. So that's the vast majority of the types of biomolecules. I'm not going to throw water in there because water is obviously not a specific biomolecule. Okay. So some of these things we can eat, and some of these things we can make, and some of them we have the ability to eat and make them. So an example of that is cholesterol. So we can make our own cholesterol. We make it from acetyl coenzyme A, which you've probably heard of before in your biology courses. So the biosynthetic pathway to making cholesterol is like 30 steps. It's quite large to make all of these ringed systems that go into making cholesterol. We can also eat cholesterol, and if you eat enough cholesterol, you basically shut down the synthesis because you've eaten enough of it. Okay. With proteins, we have the essential and non-essential amino acids, which we kind of talked a little bit about back earlier in the semester. So it's entirely possible that you are eating enough of certain amino acids that you're not having to make them, or having to make many of those, but you might need to, and you can make some of them, but others you can't. So your body has a really good way of controlling the reactions that it's doing into making and kind of negotiating all of these different biomolecules. Kind of hope that makes sense. So one of the things that I need to talk about is the concept of metabolism. And this is a word that we use a lot in just kind of everyday speak, right? Like Aunt Gertrude has a slow metabolism, that kind of thing. And some people probably realize that your thyroid has something to do with that. Like if you're hypoactive in your thyroid, you might have some problems with your weight because your metabolism is very slow. These are things that you've probably heard of and have some concept of. What you probably, well, at least what I need you to get out of that is that metabolism is actually kind of broken into two different types of reactions. So there's anabolism. So think of like anabolic steroids are building up muscle. So I'm so sorry for all the background noise. To be fair, this morning when I tried filming this the first time, it was absolutely as quiet as the dead in here. And now the dogs are just excited because it's later on. Anyway, anabolism is like, I just always remember anabolic steroids build muscle. So anabolism is the half of metabolism that's building up molecules. So if you want to think about a pathway that you're probably really familiar with, photosynthesis builds up sugar, right? That would be an anabolic pathway. There's also catabolism, which is the opposite, which is breaking down molecules. So a good example of that one that you would know about would be glycolysis. And I'm saying sugar generically here, but you can think of it specifically as glucose if you want. Okay. By the way, the lecture that I'm doing right now is just basically going to be a lot of words. It's one of those days where I just have to kind of give you a lot of words. I don't know if I said that already or not, but okay. Another word is metabolite. Okay, so a metabolite is a molecule that is kind of part of a metabolic pathway. Let's see. The one that I think most of you would probably have Watched my husband walk through the dining room with a syringe in his hand. It's time for Red's insulin, but he looked really nefarious when he did it. So, also, he still can't hear me because he's still wearing headphones. Anyway, so metabolites are molecules that are part of a metabolic pathway. And the one that I think you guys have probably heard of would be like, I think like rubisco, if you've ever heard of that molecule in photosynthesis. I don't teach photosynthesis very often. Most of my students don't want to have anything to do with plant biochemistry, even though I find it absolutely fascinating. But, right, we have all of these different molecules that, in a lot of ways, some of them have nothing to do with your body and how it runs, other than it's helping to get from point A to point B. Okay, and so we call those metabolites. Some metabolites are useful for other stuff, like they can go into multiple pathways, but it just kind of depends. So I also need to talk about a metabolic pathway. A metabolic pathway is a very defined set of reactions. So these are pathways that you have likely heard of before. So there's the light reactions of photosynthesis, and then there's the dark reactions of photosynthesis. Those would be kind of two different metabolic pathways. They basically have a defined set, like a beginning and an end. For cellular respiration, you've got glycolysis and the Krebs cycle and the electron transport chain, right? So you learn these kind of big words, and you think of them as this kind of complex process because they are. You could have a metabolic pathway that is 3 reactions. You could have one that's 30 reactions, okay? But whatever it is, some scientist somewhere, probably a consortium of a variety of scientists, have gotten together and said, this is its own distinct metabolic pathway. It goes from point A to point B. And these are the reactions that are involved in that. So hopefully that kind of makes sense. As I said, we have these different metabolites, and metabolites are going to be part of these metabolic pathways. And those metabolic pathways can be anabolic or catabolic. So if you have any questions on those words, please ask. You can hit me up and ask me. Okay. Now, I need to talk a little bit specifically about metabolic pathways. And we're going to see more of this. Oh, for the love of God, can you guys just like settle down? I love my dogs, but they're so loud right now. Having tile floors is great until you're trying to do this. Thank you, just lay there. Okay, so a metabolic pathway is a defined set of reactions. And so there are a couple of things that we need to talk about as far as a defined set of reactions goes. So I'm using Microsoft Paint for this because that's the easiest way for me to draw structures, but there's not like a bulleting indentation type of thing here. So, oh, maybe there is. No, I don't think there is. I don't know. It doesn't work the same way as in like Word or something. So I'll be using dots for bullet points and kind of arrows to indent. Okay, so one thing to know about the reactions for a very specific metabolic pathway is that they are typically clustered together. Why is that not typing? What's going on here? Yeah, it'd be so much easier if we could just do this in class. All right, I'll try that again. Okay, so the enzymes are generally found in, how want to say this? A close proximity to each other. So there are a couple ways that your enzymes can be found in close proximity. In eukaryotes, we can actually have them clustered into a specific organelle. So they can be clustered into organelles if you have them, right? So bacteria don't have organelles, but they also tend to be very small. So a good example that you probably think of when you're thinking about a metabolic pathway that is clustered in an organelle would be the TCA cycle. So I can guarantee every single one of you at some point, let me see here, have seen something that looks like this. You've seen our mitochondria jelly bean, and then you've seen the words like TCA inside of it. So that's because all of the reactions for the TCA cycle are happening inside the mitochondria, okay? So that's just one example. The light reactions of photosynthesis are happening in the chloroplasts, right? In that specific organelle. They're not happening in the nucleus, for example. So that is one really important way that we tend to cluster these reactions together so that the enzymes can work quickly by having access to basically everything that they need access to. The reactions would go by much slower if you had to factor in like transport and movement from one part of the cell to another. Okay, so we've evolved to basically couple these all really close together. Another way that we can do that is through quaternary structure. So if you remember quaternary structure, we talked about proteins, like a functional protein unit can sometimes have more than one polypeptide that makes it up. So specifically, one of the examples I gave you was the proteasome, which had 4 rings with 7 proteins each, plus these 2 end caps, right? That's a lot of protein, basically protein units to do the same thing. We will see some examples of quaternary structured proteins that work as a unit to do multiple reactions together, okay? So sometimes when we're talking about things like bacteria, we're talking about complex metabolic pathways that are happening in an organism that's one cell and doesn't have organelles to kind of segment these different reactions. So in the cases of bacteria, their reactions are all right in the cytosol because there's not really another way, but they do kind of have cellular anatomy the same way we do. They just don't have membrane-bound organelles. So you can kind of still think of them as being able to kind of couple their reactions in generally defined areas, but we're not going to talk about that at all in this class. I do tend to focus predominantly on mammalian biochemistry because so many of you are like pre-med, including Stephen and Drew. So many of you are, interested in medicine or even veterinary medicine, right? Where you're predominantly going to be working with mammals. So that's where I focus our conversation. We could equally pick up plants and learn the same type of biochemistry. I just picked this one because it's the one that you guys seem most interested in. Okay. Something else about metabolic pathways that you need to know is that they tend to be controlled. I want you to think of an example. And I gave you this example a few weeks ago, actually, where you can have branch points, right? So your body uses glucose to do a lot of stuff. It uses it to make ATP through aerobic cellular respiration. It is used to make lactic acid when you're talking about anaerobic respiration. You can use it to make glycogen if you are a mammal, or if you're a plant, you can make starch, right? That's another thing that could happen. You use it to make the ribose that's in RNA and DNA. Okay. So those are, oh, and you can use it to make fat. And you can use it to make cholesterol and steroids. So all of these pathways feed into each other and have these branch points. So at some point you have to have some level of control of each pathway. So when we define what a pathway is, there's got to be some place in that set of reactions that gets regulated by your body in some sort of way, okay? Oftentimes, this is going to be often through allosteric regulation. So you have feed-forward activation and inhibition. We talked a little bit about this, I think, in class a few weeks back. I can tell you that we tend to regulate kind of the same reactions over and over. So one of the types of reactions one of the types of reaction or places that we tend to regulate is the first step of a pathway. Like if your body, and just think about if you were engineering an organism for the first time and what you would want to have done, okay, to make this work. The first step of the pathway, when you're like, okay, do I make sugar or do I make fat? Do I make energy or do I make DNA, right? Those are very different types of pathways, but they all kind of start from the same point. So the first step of a pathway is a really good one that your body will oftentimes regulate. Another one is steps with a high energy output. So a very negative delta G. I'm going to have to do a little D and G for delta G, sorry. Which also oftentimes coincides with the ones that use ATP. So you obviously don't want to go through all of cellular respiration to make ATP to just waste it on reactions that don't need to have happen. So that's another place that tends to get regulated. Branch points. And we'll see a really good example of this when we go through glycolysis. So, you know, that point where, so for example, I said that glucose is used to make all these different things, right? The third step of glycolysis is where you go from, you go from fructose 6-phosphate to something else, okay? And it's at that point you have to commit that molecule to the rest of glycolysis, or it won't be able to go in back to make glycogen. You won't be able to use it to make DNA and RNA sugars. So those branch points where you can kind of branch off and go to different metabolic pathways, those are really, really important areas to regulate as well. Now, it's not like this for every reaction. Like we evolved these, right? So it kind of depends on which reactions you're looking at, okay? But these are just kind of general trends. So I need to take a second and give myself more area to write. because I'm going to be spending the majority of our biochemistry class talking about about carbohydrate metabolism, specifically the breakdown of glucose, there are some words that I think are useful for you to know. They're words that you'll kind of see pop up over and over again, and they are kind of words that you only really see in biochemistry or molecular biology type fields, mostly biochemistry though. So one thing is that if you're talking about carbohydrates, another word for that is saccharides. And then all of them have the same formula. They are some sort of number of carbons and an equal number of units of water attached to that. So carbon, hydrate, water. Carbohydrates, it's some number of carbons with waters attached to them. I don't know if you've ever thought about the word before, but there you go. A way that this tends to get abbreviated is CHO, is an abbreviation. It also is, true story, the abbreviation for a very commonly used cell model in toxicology and molecular biology research of Chinese hamster ovaries. So context is very important. CHO can either mean carbohydrates or it could mean Chinese hamster ovaries. I found that out in graduate school the hard way by making a fool of myself. And I know you guys are sitting there thinking, no, she would never do that. Okay. So we identify carbohydrates in various ways. So one is by the number of, and really I'm talking about monosaccharides here. So you can have heptose, which is 7 carbons. You could have a hexose, that's 6 carbons. You could probably guess, good Lord, pentose. You could probably guess what most of these are going to be. So 5 carbons. Tetrose, it's 4 carbons. And then triose or triose is 3 carbons. We don't really tend to look at carbons outside of that range for reasons that I think I'll just skip over since we're having to do everything online. But if you think about it, back when we're in organic chemistry, I talk about how certain ring structures are more stable than others, things like that. So like 5-member ring, 6-member ring, cyclopentane and cyclohexane are the most stable. If they get too big, they start getting floppy and form smaller rings. If you didn't have me in organic, you might not know that. I don't know what Dr. Matthews and Dr. P taught. Okay. So what I'll point out, and this is kind of important, this doesn't tell you the structure. Like, I can't tell you which sugar you're looking at if I say you're looking at a hexose, because there's about 1,000 different ways to have a hexose. All that does is tell you how many carbons there are. So for example, glucose, fructose, and galactose are all 6 carbon sugars. They're all different. There are several pentoses. There are multiple trioses. Dihydroxyacetone and glyceraldehyde are both triose sugars. So we just kind of need to understand that that word, that those words are just telling you how many carbons are involved. I'll also point out that it's also not telling you anything about the stereochemistry, which is very specific for sugars as well. So the next thing I want to talk about is, oh, and I'll point out OS, put that up here, Ose is a common suffix for sugar. So a lot of the words that we're going to see have ose at the end. If you went through and just plugged in any number for that X, you would find that a sugar has 1 unit of unsaturation. And so all sugars have 1 unit of unsaturation. Thinking back to organic chemistry, that means they either have a double bond somewhere or they have a ring somewhere, okay? So the interesting thing about sugars is many of them can exist in both a linear form or a cyclic form, okay? Most of you have seen cyclic versions of glucose only, but they actually exist as a cyclic form and a linear form. So all sugars have 1 unit of unsaturation. In a linear form, they can have either a ketone or an aldehyde. So if it's a ketone, then we call that a ketose sugar. And if it's an aldehyde, it's called an aldose. Okay? So for example, glucose is an aldose sugar. It has an aldehyde on it when it's in the linear form. Fructose is a ketose sugar. Okay, and we'll see examples of that. Third one is to know about the stereochemistry. There are a lot of chiral carbons on most sugars. And so the stereochemistry of those is very important. Your body will only recognize one form over another when you're talking about like the whole, all the stereoisomers combined. You only got one that it will use. So a lot of our names in biochemistry are specific to a very specific stereochemistry. Okay? So essentially you could have the R and S form of glucose, but if you flip 2 of those groups, you no longer have glucose at all. You call it another sugar name. So, and that's just the way biochemists did things a long time ago, so that's still the way that we do it now. So I talked about biochemistry using the D and L system for stereochemistry rather than the R and S system. I talked about that just a little bit when we were looking at amino acids. And we talked about how amino acids are all the L stereochemistry. All sugars are D stereochemistry. And that's the relative stereochemistry relative to glyceraldehyde as the standard that was done a billion years ago. Sorry, I'm using a new keyboard. There are some examples in nature of some organisms that can use L sugars. Again, those are like weird sulfur vent bacteria type stuff. Okay, so I want you to think of everything we're ever going to see for sugars is going to have D stereochemistry, or it's not going to work at all. Hi, Poppy. So the next thing, I'm going to see if I can copy and paste this over. Yes. I need you to remember back to organic chemistry. And there are things called Fischer projections. And what I need from you, Drew, Stephen, whoever else is with me right now, I need you to tell me if you have seen these before. I know the students I've had have seen Fischer projections before. Your organic chemistry book does not cover them until like chapter 25. It has a section on carbohydrate stereochemistry, and so they do Fischer projections then. I cover them early on when I'm doing stereochemistry and organic 1, but not everyone does that. So, Okay, Drew, you've seen them? Awesome. Appreciate the feedback. Okay. Yes, fantastic. So I'm not the only one that teaches it. Okay, so Fischer projections are, in your organic book indicates this, right? Because they're like in the chapter on carbohydrates. That's where we tend to talk about Fischer projections. They're not used a whole lot outside of biochemistry, but they are used very often when looking at sugars and amino acids, both of them. So if you look at this little picture that I copied and pasted into here, and write them down, but you don't need to memorize these structures, okay? But what I want you to see is that, let's see here, where's my pen? Let's do red. So you'll notice here on the on the fructose, what is going on? Oh, I didn't hit my, there we go. The fructose here has, well, it's got 6 carbons, 1, 2, 3, 4, 5, 6. And it's got a ketone in the middle. Glucose has an aldehyde. Mannose, another sugar that you might be less familiar with, also has an aldehyde. So we could say fructose is a ketose hexose, meaning it's 6 carbons with a ketone in the middle. Glucose and mannose are both hexoses, and they're both aldoses, meaning they're 6 carbon sugars with an aldehyde in their linear form. You'll notice, remember that Fischer projections do indicate stereochemistry, so it might be useful for you to just kind of look back at that. I didn't build any models of that. I did get to teach Organic 1 today about stereochemistry using models online, which was very weird. But I think if you just kind of refresh yourself on those a little bit and ask me any questions you have, that's probably fine. It's helpful if you can look at these, though, and just kind of know what they mean, right? So the ones that are up and down are pointing behind, and the ones out to the sides are pointing forward. Okay, so if you'll notice the difference between glucose and mannose, the CH2OH at the bottom's the same, these two are the same, and then this one's the same. The only one that's different are the OH groups at carbon number 2. If I can even like highlight that. So, you know, how would you compare D-glucose and D-mannose? Those would be enantiomers of each other. They're non-superimposable mirror images of each other, but there's also a lot of diastereomers that could be involved, right? That are not, that would be non-superimposable, non-mirror images and stuff like that. Okay. All right, so that's just kind of what I need you to know about the Brandon's finally done cleaning the kitchen. I won't complain that he's been cleaning the kitchen, but I will complain that it was so loud. Okay. The last words that I kind of need you to know are Purnose and Furanose. So if you remember back in organic chemistry, one of the common solvents that you saw a lot was THF, tetrahydrofuran. Furanose are sugars that have a furan ring. Purnose sugars, or pyranose sugars, I've heard it pronounced both ways, have a pyran ring. Those are some of those heterocycles, carbon rings that have a non-carbon atom in them, oxygen, nitrogen, sulfur, something like that. Hi, Twinkie. Are you going to make it, bud? You're acting like you're dying. He doesn't like the dishwasher. He starts to fuss, so if he barks really loud, I'm really sorry. It'll be louder for me than you, and it'll be miserable for all of us. Okay. All right, so a Purnose is a 6-membered ring that has an oxygen in it. And a furanose, or a furan, is a 5-membered ring with an oxygen in it. So some sugars are pyranose, some sugars are furanose. Most sugars can be both, just kind of depending. Okay. Let me stop for a minute and just see if there are any questions. I realize these are just a bunch of words, so at this point it should be pretty straightforward. This is one of my more boring lectures, so I'm kind of glad this is the first one we had to do online. But if you have any questions, now would be a good kind of stopping point. There's about a 1-minute delay between when I talk and when you hear me talk. So if you notice when I ask a question and then I pause a while, that's because it'll be at least a minute before you hear me say it and I start to see you guys respond. Okay. All right. What is D-stereochemistry? So D is the one that is relative to D-glyceraldehyde. Okay. In amino acids, or in a 1 chiral carbon molecule, you can kind of think of D as being synonymous with R stereochemistry, and L is more synonymous with S stereochemistry. But in sugars, because there are so many chiral carbons, it's really hard to tell. The D just means that it rotates plane polarized light the same direction that D-glyceraldehyde does. So hopefully that kind of makes sense. I mean, without getting into a lot of details, that one's kind of abstract. It's mentioned in your organic book in chapter 5 at the very beginning of the book. Fortunately, I teach both classes, so I know exactly where it is in your book if you still have it. You don't, if I told you to draw the D version of a sugar, that would be pretty impossible to do if that's kind of what you were wondering. Because in the old system, each of these combined would give you D stereochemistry overall. Whereas now we would actually go through the 4 different carbons that are chiral on that molecule and identify which is R and which ones are S. So the system that we use now is much more accurate and much more detailed. It gives you more details. But in the old days, they were like, that one looked like the way glyceraldehyde does. And so that's how they designated those. Okay. But I wouldn't ask you, I wouldn't be like, here's a sugar, draw the D isomer. You wouldn't be able to do that without having it already memorized. Okay. So hopefully that, if you're like, good Lord, I don't know how to do that, that's okay. I wouldn't expect you to. I can't do that. It's impossible. Okay. So I think I've had that on the screen long enough that I can delete it. If that's a mistake and you guys were still writing that, I'm really, really sorry. I have one more thing I want to talk to you about before we're done for the night. And I have like 15 minutes, and so I think that's good. I need to talk about the linear, oh goodness. My brain keeps getting confused. I have a keyboard and then I also got a nifty writing pad to try and do things a little bit easier, but I keep thinking it's like a mouse touchpad and it's not. So my brain doesn't like that. Okay, So I need to talk about structural forms of sugar. Okay. So if you have a 3-carbon sugar, a triose, any ring that you could make would be at maximum 4 atoms big. With sp3 hybridization, that would make those bond angles about 90 degrees, right? That would have lots and lots and lots of angle strain or ring strain. So if you're thinking, if you're thinking about 3 carbons and an oxygen, right? That's not going to be stable. And I would hope everyone in this chat would look at that and say, no. In theory, you could have 2 carbons form an epoxide. We also know those aren't particularly stable, right? So these aren't things that you're going to see happening in your body, but we can get the 5-membered rings and 6-membered rings very easily. It just means you have to have enough atoms to do that. So generally speaking, when you're talking about these structural forms of sugars, if you have anything that is 5 or 6 carbon sugars, well, I'll say greater than 5 carbon sugars, can cyclize into rings. And I'll remind you that those type of rings are called heterocycles because they have a heteroatom, meaning a non-carbon atom in them. So the 4-membered ring and the 3-membered ring, that's going to be a hot no. But 5 and 6, the furans and the pyrans, are fair game. Okay. And so now I need to remind you, not just the Fischer projections, but if you had me in class, you might remember Haworth projections. If you haven't had me in class, you've definitely seen them, even if you didn't realize it yet. So a Haworth projection is where you have a ring. So I'll draw cyclohexane as a Haworth projection. We tend to draw them kind of elongated. So normally if I were to draw cyclohexane, I would try and make, gosh, that's really hard to do on this thing. I would try and make my bond angles or bond distances about equal. But in a Haworth projection, you kind of spread them out. And you can use this to indicate stereochemistry, right, by pointing up and down. So one's pointing up, one's pointing down, right? If you were to transfer this into a chair conformation, I have one pointing up and one pointing down, right? So a Haworth projection does indicate the directionality of where atoms are pointing, which gives you kind of an idea about the stereochemistry. Biochemists, in general, don't use chair conformations. And I'd be very surprised. I don't think I've ever seen a biochemistry textbook that has them in there. Certainly not in molecular biology or cell biology. If you find any, let me know. I'd be curious to know. But in a general speaking, we only use Haworth projections. Twinkie, calm down. Please don't bark. So when we're looking at a molecule like this, we're having to remind ourselves that we're looking at it kind of implied that these bonds are kind of bolded, so they're sticking out at you. Twinkie, go lay down. Don't you dare bark at the dishwasher. I'm busy. Go lay down. So you're kind of looking at it head on, right? That's kind of how that's implied. You don't always see it with the bolded lines like that. But you usually do see them elongated. So you wouldn't see them drawn that way without the kind of elongated bonds on the top and bottom. Okay. So as far as Fischer projections and Haworth projections go, I kind of drew, let's see, I drew one of these ahead of time. It wouldn't take long. Hey, Brandon. Twinkie's very angry at the dishwasher and keeps her roughing. Can you please do something with him? Thank you. That is not spectacular resolution. Sorry. It looks much better on my screen, but I hope you can kind of see that. So this is actually glucose, and I'm showing you how you can cyclize it. So essentially you would have something brighter. You can see a little better on the white. Okay, so you could have a base basically come in, take off a proton, and then this bond breaks, grabs that carbonyl, flips up and grabs a hydrogen, okay? So that is how we cyclize. The thing is that when that oxygen is attacking the carbonyl, right, if you're thinking of a line, it could either go this way to attack, or it could go this way to attack. When that happens, you have two different versions of glucose that you can get. And if you've had cell and molecular, you've probably learned about this. You'll notice that in this example, the OH is pointing down, and in this case, the OH is pointing up. Okay, these are the alpha and beta forms of glucose. These are called anomers. Which is a type of isomer that is specific really to sugars. We just call them anomers. And when you have, they're like chair conformations, right? If you have one, you have both because they're flipping back and forth. In this case, you have both because they're flipping back and forth. They're in equilibrium, as indicated by my equilibrium arrow right there. They're in equilibrium between the linear form and then the ring forms. So we call these the alpha and beta forms of glucose. Okay. The way that I tend to show this, and I'm not the only biochemist that does this. This isn't necessarily a Dr. Mannism, but it's not something done everywhere either. So if I'm drawing glucose, and I'm drawing it in my Haworth projection, I'll keep my stereochemistry correct and identified on all these. For glucose, I go up, down, up, down. It's easy to remember it that way because it just alternates. And then here, this we can use that squiggle line that means variable stereochemistry. The stereochemistry could go either up or down. So this is a way of kind of showing both of them. The too far. So I think that I'll actually stop there. But what I want to do is give you guys kind of like a thought thing, you know, to kind of do before Wednesday. So what I want you to do is to draw the chair conformations. Look up which is alpha and which is beta. So Alpha will have 2 chair conformations. Beta will have 2 chair conformations. I think it's just good to flex your brain and remember that because it'll show you which one is more stable based off of the fewer 1,3 diaxial interactions. So draw the chair conformations, look up which is alpha and which is beta, and then learn the mechanism for becoming linear and becoming cyclic. And now I'm realizing I haven't had my chat screen up for like 10 minutes, so if you've asked me something, I didn't see it. Okay, I did not. So I'll just stop there. I got almost to where I normally do in this lecture, so that's a good sign. Hopefully what I just told you guys made sense. I know it was pretty straightforward. I basically have these notes typed out so I can post those on D2L as well, but they're all the same thing I've basically been posting, been typing as we've been talking. So let me know. I'll just sit here for an extra few minutes if you guys are curious and you have questions and you need more information. I'm happy to answer questions. Hopefully this worked okay for you. I'm really sorry it's finally quiet. Brandon stopped working in the kitchen and the dogs finally left the room, so I'm really sorry about that. I've heard of some professors who are having to do this now, but they have kids at home and says like, Mom, Dad, and they keep wanting help. I can tell you about the super scary story that happened to me. I almost died this past week. At least that's what I like to think. It was super scary for me. Red got loose at the cabin. He's old. He's 10. He's insulin dependent. You know that. We let him out to pee and he jumped off, which is really uncharacteristic for him. And we couldn't find him in the swamp. So I put on my chest high waders, like what fishermen wear. And I went out looking for him. Alpha glucose has 2 chair conformations. Yes. So each isomer would have the 2 different chairs. So alpha has 2, beta has 2. Anyway, so we're out looking and couldn't find him. And he was gone for 7 hours. It was the scaredest I think I've ever been. And when I was out looking, I almost stepped on a water moccasin / cottonmouth. I was about 1.5 feet away from it. They have an amazing defense system because they have that bright white mouth, hence the name cottonmouth. And otherwise they just kind of look like a brown turd in the water kind of thing. Like they don't really stand out. They look like a stick almost if they're stretched out. And if it hadn't gone, then I wouldn't have seen the white and I probably would have stepped on it. So that's the story about how Red almost killed me. He did come home. He showed up at some dude's yard. I keep his phone number on his collar. Always do that. Saved him. In the time since you last saw me, look at that guy. My thumbnail almost looks like a real thumbnail again. That's exciting. I miss you. It's going to be a weird semester. Okay, well, if there's no more questions, we can go. I'll post this online so you guys have it. All right, Twinkie's getting ready to bark at me. I will talk to you guys later. Have a good night, and I will chat with you again on Wednesday.